Good afternoon and welcome to the Internet of Water's peer-to-peer network webinar. Today we're talking about water data for COVID, and this is part 2 of a series on COVID wastewater surveillance data. If you'd like to view part 1, you can visit our webpage, internetofwater.org, and in the peer-to-peer network page, you can find links to our past webinars. In that webinar, we heard from our friends in New York and California and Utah who shared information about their state's efforts toward wastewater surveillance for COVID. And today we have friends from California, Wisconsin, and the CDC talking about the data infrastructure behind these programs. Before we get started, we have a few housekeeping just to keep up with. My name is Ashley Ward, and I am the Engagement and Outreach Associate with the Internet of Water. This webinar is part of our peer-to-peer network, which is a network community of practice for those of you working to modernize your water data infrastructure. And we'd love to have you join us. If you'd like to join the peer-to-peer network, please navigate to our page, internetofwater.org, and click in the top menu on the peer-to-peer network, and you can find out information about the network and how to register and become a part of the network. We'd love to have you. Also, want to remind everyone today that we are recording. The recording of this webinar will be posted on our website and distributed via email to all of you who registered for the webinar. I also want to remind you that if you have questions for our presenters today or our panelists, please place your questions in the webinar's Q&A box. I will read your questions aloud and have the panelists answer your questions. However, if you're having administrative questions, you can't hear or having a problem seeing the slides, please place any questions for our staff in the chat box, and one of us will respond to you. Now, we also have a few people participating today via WebEx. So if you're participating via WebEx, you can post your questions and any concerns or issues you're having in your chat box, and one of our staff will transfer those over to the webinar today. So again, we thank you for joining us. One of our first speaker of the day is Wiley Jennings. Wiley is a health scientist with the Waterborne Disease Prevention Branch and the National Center for Emerging and Zoonotic Infectious Diseases at the U.S. Center for Disease Control and Prevention. He serves as the data lead for the National Wastewater Surveillance System, which launched in 2020, and holds a PhD in environmental engineering from Stanford University. So we'd like to welcome Wiley. And before we do, we'd like to ask for you to complete a short poll, which you should see on your screen at this time. So Wiley, let's give everybody a moment to complete the poll, and then we'll move forward for your presentation. We have just a few people have answered. Here they come. Wiley, can you see the question?
Yep, they popped up on my screen.
Okay, great. What is your degree of familiarity with the data flows for CDC's National Wastewater Surveillance System? It looks like most people don't have a familiarity with the system. If you're participating, how are data being transferred, collated between? Quite a few are in the others category. I think we might have all that we're going to have.
Great. Thanks for running that, Ashley.
Sure. Exit this. Here we go. I'll turn it over to you, Wiley.
Great. Thanks very much. All right. Can everybody hear me okay? All good. All right. Good afternoon, everybody. Thanks for joining, and thanks to the Internet of Water folks for putting on this webinar. So I'm going to provide an overview of CDC's National Wastewater Surveillance System, which seems like it'll be good background since most people are relatively new or don't know very much about the system. So the National Wastewater Surveillance System, NWSS, we pronounce it NEWS. So apologies for the acronyms, but bear with me. I'm going to be saying NEWS a lot. The presentation is going to emphasize the data flows and the news data platform. So I also have some slides on data analysis, which is a key part of the news data platform, but I'm going to jump through those pretty quickly so that we can keep the focus more on data flows and data infrastructure, which is really the focus of this webinar. Next slide, please. Great. So this map depicts the states in which surveillance at least one treatment plant in the US is being conducted, supported by federal funds. So the 15 states in green and dark blue are states where the public health department or a local health department within that state is directly engaged with news. That's either through participation in regular meetings to support wastewater surveillance implementation or through actual data submission to news. News is currently receiving routine data submission from 5 states, representing over 5,000 wastewater samples from more than 160 treatment plants. We continue to work closely with other state partners who are at various stages of surveillance implementation or data submission to news. Then the other states that are highlighted in light blue are states where wastewater is being sampled through a public-private partnership administered by the US Department of Health and Human Services. And that's collecting wastewater samples, or was collecting wastewater samples through February at 105 treatment plants across the country, covering approximately 10% of the US population. So this public-private partnership is expected to expand in April and May, and we continue to work with HHS to try to integrate those data into the CDC news platform where they can be turned around and then used for public health decision making. Next slide, please. So the National Wastewater Surveillance System, or NEWS, is a US CDC and Department of Health and Human Services led effort to support the use of wastewater as a medium for public health surveillance in the US. NEWS supports wastewater surveillance data collection through laboratory and epidemiology capacity, technical support, and funding. And then it collates, harmonizes, and analyzes those data for use in public health response. Additionally, NEWS organizes peer-to-peer community of practice groups for public health departments who are implementing wastewater surveillance and public health laboratories conducting or considering wastewater testing. Currently, NEWS is focused on SARS-CoV-2, but NEWS is envisioned as a flexible multi-target platform for other health targets as well, including infectious agents like enteric, viral, and bacterial pathogens, and non-infectious targets like antibiotic resistance genes or opioids. So this slide provides a high-level overview of the components of the National Wastewater Surveillance System, which I'm going to walk through. So starting in the top left, communities contribute wastewater to centralized sewage collection systems. That wastewater flows to a centralized treatment plant where wastewater samples are collected, representing a pooled community sample. NEWS is primarily focused on this community level surveillance where samples are collected either from treatment plants or interception points in a centralized sewage system. However, the system can receive data from targeted surveillance efforts, such as from correctional or long-term care facilities. So these wastewater samples are then transported to laboratories for analysis. Currently, there are a wide range of labs participating. Initially, most of the labs running this testing were academic, but increasingly we're seeing commercial and public health labs come on board, which makes a lot of sense for the long-term sustainability of the surveillance system. Data from the utilities and laboratories are then collated by public health departments, typically at the state level. This step involves a tremendous amount of work for all parties, the health departments, the labs, and the wastewater utilities. And there really is a large diversity of approaches to organizing this information flow across the US. And it's one of the key bottlenecks in the speed and sustainability of this surveillance system. So as the news team staffs up over the coming months, we hope to be able to provide more technical assistance and tools to support this aspect of the data pipeline. It's been great to hear that the Internet of Water is also thinking about how to support state level data collection and collation. So certainly I hope we get to talk more about this in the panel discussion. So CDC news staff then worked with the state public health department staff to validate and standardize those data for submission to the news database, which is hosted on the Decipher platform, which is what I'm going to spend a lot of this presentation talking about. So Decipher stands for Data Collation and Integration for Public Health Event Response. And it's a secure integrated platform for health surveillance information hosted at CDC currently. Those data are then normalized and analyzed and returned to health departments as a dashboard on that platform and as downloadable analyzed data. We're also working to make these data publicly accessible by pushing a dashboard to the CDC COVID data tracker webpage and setting up a process to facilitate public data requests and approvals for paired wastewater data and sewer shed level case data. And that both of those efforts are in the works and have been in the works, but continue to be ongoing. Next slide, please. So complete submission to news comprises 4 data elements. The first element is the wastewater metadata and SARS-CoV-2 testing results. And this includes information from the utilities, describing treatment plant operational features, sample collection methods, and then laboratory methods and quality control measurements. So these data can be submitted to Decipher by a health department representative who logs into secure news platform and then uploads a CSV file. Wastewater data are really the heart of this system. So only this first element is really required to have data analyzed and shown in the dashboard. The remaining elements 2, 3, and 4 are important for interpreting the data spatially and for contextualizing wastewater data with case data. These additional elements, however, do add considerable complexity to the data collection and collation effort. For example, the sewer shed spatial boundaries are used for mapping data accurately, since wastewater data do not represent populations that correspond neatly to common administrative boundaries like counties or towns. And they're also used by health departments to subset their case data so that they accurately represent the population contributing wastewater to the sampling location. In the panel discussion is, to our knowledge, there has never been an effort to standardize and collate these sewer shed boundary data at the state level, and certainly not for use in disease surveillance. And this process can really be quite a significant effort, both to generate these data, since many utilities don't have them or don't have updated ones, and then to organize and manage that data. So this is really another place where we should be thinking about how we can improve data flows. So in the platform, we then plot the wastewater data alongside those case data. And we also request, element number 4, we also request laboratory protocols, which kind of can help to fill in the methodological nuances that aren't captured in the CSV data submission. Since we know, this is a continually rapidly growing and developing field where the methods are quite variable across the country and continue to evolve. Next slide, please. So in the next couple of slides, I'm going to provide an overview of the Decipher platform to help give a sense of what's possible on the platform and what the actual data submission process looks like. So Decipher, which is built on Palantir Foundry software, is built as a one-stop shop for data submission, management, and analysis. The benefits of an integrated system include that it cuts out the data translation issues between the database, other databases that are hosted on this platform, and then the data analysis steps. And it makes for easier troubleshooting since the code and data are in the same environment, and facilitates QC on the platform by both submitters and CDC staff. And now this kind of emphasis on that translation between database and code for analysis is perhaps somewhat unique to a surveillance system, where really the end product is some sort of visual product or data product that can be used to help guide public health decision making, rather than just merely serving as a database or a data repository. If you could please advance, I think some bullets should show up. Great. Yeah, you can just go through the whole list of them. Thanks. So I'm going to talk through a couple of these bullets and then skip over most of them. But the first one, Decipher makes merging of multiple data submission flows possible. So we're not really taking advantage of this feature directly for wastewater data collection at the moment, but it will likely become useful as we work towards more modern data submission and ingestion processes. So on the data ingestion, and I should note here that CDC security does not allow automated pushes of data from external databases, but does allow CDC to set up pulls from databases. So setting those automated procedures up is something that we hope to work towards over the next year plus for news. So the second point here is that Decipher facilitates an integration of disparate data sources. So we can set up API calls to public databases and bring in data from other federal databases, which is how we integrate the COVID-19 case surveillance data with the wastewater data. So here, I guess, note that only a few variables for the COVID-19 case data are actually submitted by the wastewater submitters. And then we link those few variables to other databases at CDC to bring in the remaining metadata, like the various dates that can be associated with case data. In addition, we are currently working with NCBI to build out a bioproject template for submission of wastewater surveillance sequencing data to NCBI. And then we hope to link these data to data that are submitted to news. Additionally, Decipher has a number of other features that add functionality for an active surveillance system beyond being merely a data repository. So it has version control and archiving tools, build scheduling tools, coding environments that support Python, R, JavaScript, SQL, Spark languages that are used for building out automated data analysis pipelines, and to produce both the analyzed data sets that can be downloaded and also to help build dashboards. It has a bunch of built-in graphical interface tools for interactive dashboard building that any of the approved users can go in and do their own data analysis and dashboard building. And then it has some more polished tools for production dashboards as well. Next slide, please. Great. So currently data submission to news can be performed through a manual form-based process or through a CSV upload process. We're actually doing away with the manual upload process at the moment. So it's moving forward, going to be exclusively through submitting a CSV file through that secure platform. This is sort of, we initially built it out as a series of forms and a relational database because this is sort of a more common data submission structure for other surveillance systems at CDC, but just based on the way that wastewater data are being collected and the frequency and amount of that data that's being collected, it doesn't really make a lot of sense for data to be entered this way. So currently we are moving forward with the CSV upload process entirely. Next slide, please. So once those data are uploaded, Decipher has a number of built-in tools, which I've mentioned, for data exploration and quality control. So it's a really nice intuitive graphical interface for querying and visualizing data and for detecting QC issues. If you could click through, there's a couple bubbles that'll pop up here. Yeah, just keep. Okay, great. Oh, sorry, I went too far. So I've highlighted in these, what used to be red boxes, but now gray boxes, some of the nice features. There's a query bar at the top. You can view things sort of in aggregation on these plots, or you can look at a line list view of the data as well. I'm highlighting here, there's obviously one erroneous date that's showing up in that right side plot. There's a test result data has been submitted, in the future. So this is a nice way to do some sort of QC that's interactive. We're also in the midst of making some changes to the way that edits in the system are done. So essentially, submitters are going to be able to make edits to their data in their source database or in the source file that they upload, and then just re-upload those changes, and it'll overwrite previous errors instead of having to do a lot of this data editing manually in the platform. We are also currently, we're sort of at this phase right now with news as a new system where we've received enough feedback from the early submitters who have been submitting data to us that we are making some changes to the system. We're working really hard to really thoroughly define value sets for these variables, which is going to enable us to implement a lot more automated QC checking and return of data validation reports to submitters. One of the limitations of the current configuration of the system is that we can't do any data validation outside the system currently. So users do have to submit their data. It has to be ingested, and then that validation is run on platform, and reports are then turned around and sent back to submitters. With this sort of kill and replace upload procedure, though, hopefully the cost is fairly low on having to upload that data first, since you can make revisions to the data in the source database and then re-upload it and overwrite that previous data. Next slide, please. So in the remainder of the presentation, I'm just going to highlight some of the data visualization and analysis that's currently implemented in Decipher, but not go into too much detail here. So this is a screenshot of the internal facing news Decipher dashboard, which contains 4 primary data elements. If you could click through 4 times, I think. 1. Yep. Okay, great. So that first one, number 1 on the top right side, is a time series of normalized wastewater levels of SARS-CoV-2, shown in black dots and with a black smooth line. And then these wastewater levels are overlaid on case incidents, which is shown as the gray bars with the light gray smooth line. The trends in wastewater data are classified into sustained decrease, decrease, plateau, increase, and sustained increase, and they're shown as those color-coded categories at the bottom of the time series plot, and then also the little boxes at the bottom. Second component of this is the map. So the sampling sites are shown on the map on the left side and are color-coded by their current trend classification. So the increase to decrease spectrum that I just described. And then points can be clicked to bring up the corresponding time series. Third, there's a grid listing the number of sites within each current trend. And that grid is expandable to show sites and metadata for those sites. And then fourth, on the left-hand side, there's a list of sampling sites where the most recent wastewater sample indicated an alert. And an alert is a statistical metric that basically says that the wastewater levels that were just measured are higher than would be expected given the recent trend in the wastewater data. And then there's a decent amount of filtering that can be done here, adjusting of X and Y scales as well to aid visualization. One of the really tricky things about these wastewater data is figuring out at scales, especially when you're comparing data across sites or over time. Next slide, please. So this is if you could just click through one, there we go, I think, sorry. This is a big topic and I'm just gonna skip right over it, but concentrations of SARS-CoV-2 RNA in wastewater may change not only as a result of changing amounts of RNA in the system, but also a result of dilution or losses in the sewage system. They may vary from day to day and from site to site. So normalizing SARS-CoV-2 concentrations for those effects is likely very important. There are some places where wastewater concentrations or flow volumes, fecal content seem to be pretty constant, and perhaps it's less important there, but it's certainly important to assess that initially at least. So the news analysis is currently using flow-based normalization. And I'll just leave it at that. So next slide, please. This slide just highlights some of the other considerations that are built into the Decipher time series visualizations. In particular, we chose to plot wastewater and case data together because the case data help contextualize the wastewater data. So it's useful to visually compare wastewater data with case data, because case data, especially for public health departments, are a more familiar metric than wastewater data, and other surveillance indicators such as percent positivity or hospitalization rates could be useful for this purpose as well. Case data are sort of the most common. It's also nice to compare the wastewater data with the case data because it illustrates that some of the day-to-day variability in values that we sort of expect to see from wastewater data are not unique to wastewater data, since it is often seen in case data as well. There's numerous reporting artifacts that make the case data, sort of a bouncy measure of true underlying community infections as well. Next slide, please. Click through. Great. There we go. So beyond the time series plots I've discussed so far, there are also two statistical metrics currently implemented in Decipher. So the wastewater trends and wastewater alerts. And the goal of both of these metrics is, one, to provide an easily interpretable yet statistically robust metric useful for public health action, and two, to be directly comparable across lab methods and sampling sites, and be applicable regardless of sampling frequency, which can vary. Now, I'll say that these metrics can be applied regardless of the laboratory methods, as long as they meet our minimum data criteria. One of the main advantages of the trend analysis is that it does facilitate that comparability of that metric across sites. So you can compare trends across sites, even if you can't compare absolute concentrations across sites. Next slide, please. So as everyone on this call knows, a unique feature of wastewater data is the geographic area that they represent. So these geographic areas, called sewer sheds, don't neatly align with other administrative boundaries. So accurately representing these requires additional spatial information, which I've mentioned before. So a choropleth map, like the one shown here, enables comparison of data across space at a given point in time, which means that that plotted metric must, in fact, be comparable at any given moment in time. And since we continue to discourage comparison of absolute concentrations across space and certainly across laboratory methods, we only show trends at each site. Next slide, please. So in addition to the trends and alerts displayed in the dashboard, we continue to develop additional metrics describing the relationship of wastewater data to case data. At the moment, these metrics are primarily for internal evaluation of system performance, and we're still determining whether they have utility for public health decision-making. So just briefly, the first metric we're developing is to try to compare wastewater SARS-CoV-2 levels and case rates. And then the second one is a metric to assess the leading or lagging relationships of wastewater data to case incidence in each sewer shed. And these are just quite difficult metrics to develop in practice because of the wide range in sampling frequency and other factors that vary from site to site. So with that, I'm going to wrap up my presentation. The last slide is just our contact information. So if you have follow-up for the CDC news program, you can reach out to us at nwss@cdc.gov. and thanks for your time.
Great. Thanks, Wiley. So we're going to switch now to our panel. And I'll if there's any questions, just remind all of you to place any questions you have for Wiley about the Decipher program or for our panelists in your Q&A, and we'll ask them of the panelists at that time. And so first, I'd like to introduce our moderator for the day, Greg Gearhart. Greg is a deputy director at the California State Water Resources Control Board and director of the Office of Information Management and Analysis. Prior to this position, Greg served as a statewide stormwater program manager, and throughout his career has worked in many program areas, including wetlands, watershed management, organizational development, and enforcement. Greg received his degree in environmental resources engineering from Humboldt State University and is a longtime friend of the Internet of Water. So thank you, Greg, for moderating today. And a brief introduction of our panelists. You all have met Wiley already, and he will be joining us on the panel, but we also have Nathan Klockow. Nathan is a research scientist at the Wisconsin Department of Health Services in the Bureau of Environmental and Occupational Health. Prior to focusing his efforts on wastewater surveillance, he assisted with establishing metrics for Wisconsin's COVID-19 response. Nathan has received a Master in Public Health from the Yale School of Public Health with a concentration in environmental health sciences, and he lives in Madison, Wisconsin. Thank you so much for joining us, Nathan. And then Rafael Maestu, did I say that correctly? Did I do it? I did it. Yay. Rafa is an economist and chief data scientist with the Office of Information Management and Analysis at the State Water Resources Control Board. Rafa joined the Water Board in 1999, and his experience includes economic analysis, program evaluation, statistical analysis, water management, water conservation, organizational performance management, environmental indicators, performance reporting, strategic planning, and information systems development. Now I have to take a deep breath, Rafa. Thank you so much for joining us today. And then we have Kyle Onda. Kyle is the data architect for the Internet of Water. He's a trained environmental engineer, urban planner, environmental public health professional, and social scientist who has spent a 12-year academic and professional career working with data, characterizing the many aspects of water, the natural and built environments, and people. He's worked on a wide range of policy questions, from local land use decisions to international infrastructure finance. He's currently a PhD candidate in the City and Regional Planning Department at UNC Chapel Hill, from where he also holds a master's degree in City and Regional Planning and one in Public Health. Kyle also has a degree in environmental engineering from Stanford. So thanks so much, Kyle, for joining us today. And Greg, I will turn it over to you to lead the panel discussion and remind all of you to please put your questions in the Q&A.
Thank you, Ashley and Lily and David and everyone at the Internet of Water. Wiley, great presentation. Thanks for laying out that amazing foundation of the work. It seems like every time I hear you talk, there's new developments and new progress being made in the data flow and the interpretation. So well done to everyone. So I'm going to start by letting the panelists who didn't have a chance, like other than Wiley, basically, to get a couple minutes to kind of frame who we are. I know that we have two different states represented here on our panel today. People might want to know a little context. You know, this isn't about the state approaches. We did that last time. But, you know, maybe start with Nathan. Could you give a quick overview of how Wisconsin, you know, generally does its data flow and how many facilities and labs and things like that? That would be great.
Sure. Yeah. So Wisconsin currently has engaged with about 70 wastewater treatment facilities around the state. The State Lab of Hygiene here has done the primarily coordinated outreach to that, to those facilities to enroll them. But basically the facilities collect their samples, ship them to either the State Lab of Hygiene or an academic lab that we're partnered with. Those two labs collect the data, and then the State Lab of Hygiene enters the data into their internal database, which automatically scrapes the data and sends it to us in an output daily through a secure file transfer protocol. We have an in-house database that we created to house the data. The academic lab sort of does manual uploads to that as they have information ready. But then once we have the data in within our database, our SQL database, we are able to manipulate it as needed for our own analytic purposes, for external presentation on our dashboard, and also for preparation to upload into the Decipher platform.
Great, thank you. Rafa, could I ask you to give a similar brief overview of California's scenarios?
Sure. You probably are in a better place to explain it than I am, but I'm going to give it a try. So in California, we started around the end of November to collect samples from five utilities, 6 systems. That is pretty much the largest one. So it's about 32% of the population of California that we are now covering. And we just start collecting samples like once a week for the month of December, and then 3 times a week for each one of the systems since January. So since then, we actually been collecting all that data. And the process is right now a manual process. They put the data into an Excel spreadsheet with all the data elements in the right order, and then they submit it to the water board, to me, with an email attachment. That's simple. And then from there, we put into a SharePoint site where the Department of Public Health actually collects the data and enters, upload it into the Decipher system. So for us, the coordination is kind of simple because we are working with 5 different managers or contact names for each one of these systems. So the communication is very fluent. It was hard at the beginning. We were seeing a lot of errors in the data format and things like that. But we were dealing with those issues on a case-by-case basis. But it's manual. It's a manual process. It's time-consuming, but I think it provides accurate data.
Thank you, Rafa. Kyle, I know one of the key principles and goals of the Internet of Water is to build out the sexy thing called data infrastructure. Everyone wants to see that thing built. That's me being sarcastic, but I'm wondering what you see from your perspective as one of the potential value-added benefits of working with the Internet of Water. I'm thinking things like persistent identifying, physical locations, things like that. But any other thoughts that you might have to sort of frame Internet of Water would be great.
Sure. So, so for those who don't know already, right, the Internet of Water is really basically focused on trying to get all this data that's collected for all kinds of primary uses discoverable and accessible for use for secondary uses by all sorts of people. And so something that's really of interest to me about this COVID data is particularly these sewer sheds, right? These are kind of like a golden nugget of data that have been really a hard nut to crack for a very long time. And now that we have this quasi-national infrastructure that's at least requesting this, that seems like a really nice opportunity to leverage the collection and then publication of that data for secondary uses. So sort of a question I have for Wiley is, I'm curious how that data is being collected and or published and what the process is for getting for downloading the publicly accessible version of that data, and if those boundaries are part of that as a start. Yeah.
Well, I think that's perfect. Wiley, did you get that? Came right at you.
Sure. Yeah, happy to take that if you want to go there, Greg.
Yeah.
Okay, yeah. No, I think that that's absolutely right. And, it's pretty clear that this is the first large organized effort to try to organize and collect these polygon files, because it's really hard. Makes it clear that it hasn't been done before. And so I guess I'll talk about the way that it's being done first, and then the second part of the question, which is the publicly accessible part of the question. In terms of the way it's being collected, it really varies a lot from state to state and from utility to utility. And Rafa and Nathan can talk about what this looks like in California and Wisconsin. One of the things that is a major opportunity and sort of an obstacle to integrating wastewater as a disease surveillance tool, which is of course CDC's angle, is that it really does bring together a lot of partners into sort of the public health data pipeline that haven't been a part of the public health data pipeline before. Our utilities are really important. Utility work is really important components of sort of our national public health infrastructure, but not typically in sort of a disease surveillance type way. So the way that sort of makes its way up to the state level or even to the local government level kind of depends on how integrated environmental folks are or water folks are into the health department. Since CDC, we really see this as a disease surveillance platform. We want our main point of contacts to be health departments. At many states, environmental health and water folks are part of the health department, but in other states they aren't. And so those interpersonal relationships really are the key to getting this data together. And when those people at the state level already have those relationships built with the wastewater folks, it makes it a lot easier. So, I mean, a key part of this is building those relationships for sure. And then obviously some utilities, especially the larger ones, already have this data available. Smaller ones don't. Some are hiring engineering consulting firms to help develop or update these data. So there really is quite a wide range, I think, out there of how easily accessible these data are and how they're being collected and how those relationships are built. In some places, they're really already very strong, so that part isn't an issue. We are requesting those data, and then I know, Nathan in Wisconsin, other folks, other state level dashboards are also using those data to represent wastewater data spatially. Utah on their public facing dashboard, I believe Ohio as well. Those data tend to be sensitive. So it's quite often that we hear that utilities don't want to make those data, the raw data, public. They can be used to represent, sort of a static image on a dashboard, but the files themselves, they don't want to be made public. And that's often sort of a precondition for participating. And so that's something that we're being very careful to honor. And so in our public data, the data that we are going to make publicly accessible, obviously we are going to suppress information that might risk sharing personally identifiable information. So we have certain COVID case numbers that we're not going to share data below. We have certain population sizes that we aren't going to share data below. So for very, very small sewer sheds, or for sampling efforts that target an individual facility or institution or building. We also won't be sharing those sewer shed polygon files that are submitted to CDC. So in terms of a kind of nationally collated data set, I think that's something that we can't offer now, but I think certainly this data collection effort is a first step towards that data being more available. And we'll see where those relationships go and where that trust goes and how that impacts the availability of those data in the future.
Thank you for that question, Kyle, and Wiley, great answer. I'm curious, have you documented those de-identification and data governance policy or processes that you just described?
Yeah, so I didn't nail down any number. I didn't specify any numbers because those exact numbers are still going through a approval process at CDC. But yeah, those data governance documents we're hoping to finalize soon. They're going through review at CDC.
And could those be shared once they're finalized?
Yeah, I think those will be able to be shared once they're finalized. They'll be, they'll be part of the data request process itself, so they will be public.
I think that would be an example of a great resource that other entities, states, and people doing this work could borrow from. So they rewrite their own data governance around a topic that's probably pretty uniform. Rafa, I'm wondering if you could speak a little bit to this polygon boundary data golden nugget. And one of the things that interests me is this, there's definitely more than just polygons. There's there's flow routing kind of questions. There's all sorts of things that we could analyze, pump stations, there's all sorts of things that would be additional data and not just a polygon. So what are your thoughts?
So I think this is actually driving a great conversation internally at the water boards for the need of these sewer sewage boundary areas. In some cases, we have that information, like for programs like stormwater or for tracking sewage spills. But when we start asking for these boundaries for these very 5 large systems, like Los Angeles, Orange County, San Diego, etc., it wasn't easy to get, and it wasn't easy to get because they didn't have it in the first place. They didn't have it defined that way. So by us starting to ask for this information, for this data in this format, it maybe it's starting to trigger the need for this to share this, actually to make this in machine readable format that is part of the reporting requirements of the data that they need to submit to the state. I think this is a great example we know there is a need for it. We have also this data for the drinking water programs. We worked for the last 3 or 4 years to define these service areas. But for drinking water, it kind of made a lot of sense. For wastewater, it wasn't really made a high priority. But because of this effort, I think we're going to start the conversation to expand the polygons, the service areas for wastewater to be required to be submitted to the state. But this is a conversation we are starting to have.
Yeah, it's almost like there's geospatial context tiers, like tier 1 might be just basic polygon for the whole collection system, or, some like Huck 14 equivalent.
Right, exactly. So it's not just the entire service area, but the water sets. So you take a sample from the collection system. What is the area that drains into that point? That's a whole new world of data collection.
Yeah, Nathan, I see you nodding your head, and I'm wondering, like, you guys, I think also have collection system data coming in too from, I don't know if it's like from hospitals or prisons or something else, but is that true that you have additional data sets, and what are your thoughts about geospatial context?
Yeah, we are primarily only using municipal systems. We don't have any sort of point systems coming in, but we have engaged with a few of our larger systems in Milwaukee and Madison who have the systems more mapped out. Basically, we know where all the pump stations are. We know the sub watersheds. We started trying to tease that apart to see how we could do upstream sampling, and we realized that it is a very, very messy data flow. Different systems will turn off entire watersheds, reroute things for an entire day into different pumping systems, only for like a few days while they're doing quick maintenance, or for a longer period if they're doing construction. Some sites on heavy rain days, they'll entirely reflow, and trying to incorporate all of those different data sources into this was daunting and probably fruitless, because we'd likely be missing something. So we basically worked with each of these systems to come up with sort of a best case scenario, best estimate for like the highest average. This is what you'll get on an average day. But that was a difficult process. The entire collection process in general was difficult because we were getting PDF maps in of planning areas. Yet you have the wastewater planning areas, the actual pipe networks. They're all a little bit different. Every treatment facility has something different that they either build in-house or they contract out for. And we were lucky to work with our GIS team here in Wisconsin DHS that really helped build out sort of a standard method for us to display things and upload to CDC. But it was a time-consuming process.
Well, okay, perfect. Back to you, Kyle. So how's the Internet of Water going to fix this geospatial boundary craziness out there?
Well, I would encourage myself to reach out to Nathan and see about what can be done to help publicize this standardized process you came up with, and see how it might apply to other states for sure. That's something we've vaguely been involved with on the drinking water side, coming up with a standard process to represent, pipe networks or various drawn maps as a geospatial polygon. On the other hand, I think it would be interesting to better understand what the sort of privacy concerns around a sewer shed polygon actually are, and understand the conditions under which that's a concern, and the conditions under which maybe there can just be more of a cultural change that we can start thinking about.
Okay, well, good. So I'm open to going where the panel wants to go, but some of the other topics that I had in mind were the always exciting metadata standards topic. We could also talk a little bit about data models, and it seems like we're, in the United States, we're evolving into this CDC-driven Decipher News data model for the wastewater data, but there are other ones out there that at least we've heard about and seen in California, like the Canadian data model around this. And I'm wondering about how conversations might be happening with making this data more interoperable within the context of the United States, but even larger than that, like nationally. Has anyone heard of anything about other data models? Or do you want to talk about metadata too? So go ahead and chime in if anyone wants to.
Yeah, I would love to hear if others have come across other publicized or not publicized data models being used internationally. I'm very familiar with the Canadian, or it's the Ottawa data model, but I'm not familiar with others, although I know that they must be out there since there are other national scale wastewater surveillance efforts. I think I'll just make one comment in sort of our approach to developing the CDC data model, which was, certainly a couple of things were prioritized. One was speed, and another one was that it could be very quickly understood and was user-friendly given sort of the IT infrastructure that was common at a lot of state health departments. So I think certainly it's not the most flexible model. It could be built to be a lot more, and it could be adopted for more relational sort of structures and a longer data format rather than, it's quite wide the way that it's structured. On the other hand, in some of our conversations, I think like the Ottawa data model is great, but also that sort of learning curve is a lot steeper to get folks onboarded to actually using it and conveying that data, but from labs to utilities, labs, and health departments. So it really is about striking that balance between a really flexible, really robust, beautiful from a data science perspective data model, and what can be quickly adopted and used and turned around for surveillance on the short term.
So did you focus on components that change a lot versus components that don't? Like what was the thought process to be a little more nuanced? I know just from watching Rafa, there's definitely, there's the flow of the lab data, and then there's like this other background data for wastewater context that doesn't necessarily have to be updated quite as frequently. Was that the thought process?
Yeah, the initial thought process was that there was some, sort of wastewater treatment plant context. When we started building this out last summer, it really was wastewater treatment plant centric. I think we've tried to make some changes since then to allow it to be a little bit more flexible and allow for things like more upstream sampling or more sort of facility targeted sampling. So yeah, there's sort of treatment plant, quote unquote demographic characteristic kind of profile. And then there's things about the sample collection process, which once you sort of develop that, you're probably going to keep your collection process pretty stable over time. And then there's laboratory processing details, again, in flux at the beginning, but hopefully stabilizes at least for a given lab and stays pretty constant over time. And then there's a couple tables that really vary, metadata for the sample, like we want flow rates correspond to each time a sample is collected at a treatment plant. And then some other water quality parameters that vary from sample to sample. And then of course the actual sort of SARS-CoV-2 quantification results and other lab QC data that go along with that vary from sample to sample.
Rafa?
So I think it's a great thing that we have a data model to follow. Even though it's trying to capture, I think they have like 74 data elements right now that we are reporting for each sample. And most of those data elements are kind of the same values every time. That's what I broken down into these are the dynamic values and the static values. But I think the real benefit of having like the CDC imposing some, like this is the format. Just send me the data in this format. It really simplified the process of communicating with the laboratories and the treatment plants on how the data needed to be provided. And the model that you created, it actually has a lot of flexibility in terms of that many of the fields, the valid values are free text. That is not really like you have to choose, from this list. That made a lot of the explaining the metadata, explaining what the data is or how the data was collected or the laboratory method. It made it very flexible enough to actually to include everybody. The other thing that I want to mention is that there's a lot of, even though there are 74 data elements, in California, we are collecting 7 additional data elements, like the phone number and the contact name of the person who's submitting the data, or the day they submit the data to us, or even the different name for the wastewater treatment plant that we provide to you. So I think there's still room for adding, data elements to the data format that that is going to be useful in the future.
I'm curious, this is kind of for anyone, but I'll go to Nathan next if you're interested. How does variant data sort of land on this ecosystem? And are you guys doing any kind of variant analysis in Wisconsin?
Well, we're in the process of sort of building that out, and we haven't quite gotten how that's going to fit into this ecosystem quite yet. There have been some, I think there have been some delays getting the actual sampling run and sequencing running up. So we haven't had those discussions quite yet.
Do you have like metadata standards for the molecular methods themselves, or how to store the variant data, even if it doesn't have a home yet?
Oh, I believe so. It's it's not distinct in the wastewater database that we have built out right now. We would need to add additional fields into that, into our homegrown database. But again, I would need to coordinate with the state lab who will be doing all of our variant analyses to see what data elements they basically see how they report that. Those are conversations that need to happen.
Yeah. Wiley, are you aware of any discussions or progress made on that whole integrating in variant data?
Yeah, certainly. I think there's a couple things. One is that, there are these sort of PCR-based single amplicon detection of mutations that are being, that are used in clinical testing and that are also being marketed and used somewhat for wastewater testing. And it's very important to draw a distinction there that those results could be reported identically to the sort of data that we're asking for right now. Because all the data we're collecting right now is we're just counting SARS-CoV-2 RNA in wastewater. So if you have another, similar PCR assay that's also just counting, it's counts of these mutations, that could be reported identically. It doesn't require any changes. The issue though is that those tests are not really applicable for wastewater variant detection. That it's quite different from when you have an individual clinical sample and you can do this sort of tiered screening of these various mutations, test for the presence of this mutation. Oh, if it's present, test for the presence of this mutation in a different part of the genome, test for the present, until you get up to enough mutations that you have pretty high confidence that you're mapping it to a variant. It's important to make a distinction between mutations and variants because the B117 UK variant is comprised of something like 15 mutations. In wastewater, it's a huge pooled sample and the RNA is in various states of degradation and fragmentation. And so detecting a single, even if we were to do 2 independent tests and detect a mutation here and then detect another mutation here, we don't have any information about whether those are coming from the same genome or whether they're fractured. So you can't use that sort of screening in the same way. So in order to do sort of variant detection or screening in wastewater, you can do some initial screening with those PCR-based tests, but then you really do have to apply some sort of longer, more comprehensive sequencing technology, which means that the nature of the data are quite different and would require quite a different sort of format and metadata, which is why we're working with NCBI to help build out what they call a bio project. So basically a template for submission to that. And they really are the experts there on that, that sequencing data, and then use their expertise as well as sort of the data cleaning. They have one of the other concerns that comes up with sequencing is sequencing of human genetic material. So especially for a public database, NCBI has tools to scrub human sequences from their sequencing results. So they can scrub those human sequences and produce only, microbial or viral or SARS-CoV-2 specific sequences, which gets at some of the PII concerns for, NCBI would be a public facing database. And then we would link to that sort of cleaned data without actually having to handle the ingestion of that sequencing data ourselves.
So this seems like a real critical step going forward as the world goes through, the next path, the iterations ahead for not only SARS-CoV-2 mutations and variances, but whatever else comes along. And so I'm curious to maintain this conversation about this, because I think that's going to be really a vital data modeling step. Is like how on one hand, yeah, it's the same kind of data, but you would at least need to indicate, that it's this variant or this sequence mutation or whatever that you're actually putting into that, and what it means. Like, I don't know, can you quantify those PCR? Sort of like, are they more binary, like it's there or it's not there, or can you use it to sort of say loading in a sewer shed? And so all sorts of questions come to mind about the data.
Greg, we have a question relative to this conversation, this topic. And we actually have had 2 very similar questions, but this is really about including other data. And for this question, what about including vaccination data from public health agencies? That would be an interesting variable when looking at trends and inferring herd immunity. So just wanted to put that out there and allow you all a chance to answer.
Wow, good one. I think, Wiley, sorry, it's going to land on you first because that's sort of cutting edge, it sounds like.
Yeah, I mean, obviously that's a great idea, and one of sort of the ideal use cases, I think, for this wastewater data is to be able to look at the impact of policy decisions, and one of those sort of policy decisions or rollouts being vaccination campaigns, obviously. So yeah, we don't currently have a clear path forward to linking data sets on vaccination percentage or sort of vaccination timelines. I think the more likely, at least in the short to midterm, the way that's going to happen is just sort of on just monitoring of the trends in wastewater and comparing those to vaccine rollout in similar areas. It's going to be very hard on any individual basis to infer any kind of causality there because there are just so many confounding factors. But it's possible that some sort of larger scale investigation that looked at I think sort of more of an economic scale national investigation that looked at wastewater data in many jurisdictions, comparing to if there's really evidence of differential vaccine rollout in those locations. There may be ways to tease that out, but we don't currently have a clear path forward to directly linking those data.
Thanks. Are there any other questions from the audience at this point, Ashley?
One question that I'm not sure if this group can address, but I'll throw it out there and we'll see what happens, really has to do with the cost of such surveillance systems. And the question comes in as, any idea of the cost per sample or, the cost to establish wastewater surveillance and the supporting data infrastructure. And is that a limiting component? I'm not sure if this is the right crowd to answer that question, but that's a hard one. So I just thought I'd pass that on.
I mean, I'll take a stab. I'm not shy about diving into this kind of stuff. I think it depends on what the project is and how people who are doing the project scope it and frame the work. I think building out data infrastructure and ecosystem kind of scenarios like we're talking about to improve data flow. One of the things that we're using in California is this idea of less than 72 hours from the time of sampling to the time of the data making its way to Decipher and News. Less than that, that would be, we would be doing well if we could get there today, but less than that would be even more important going forward, we think. And so, framing the projects that we're looking at in California to, improve incrementally that problem without solving it, it's a multi-million-dollar, multi-year IT project if you say we're going to solve that problem today. And it will take you years probably in California to build that out. But if you have some resources and you can dedicate it to incremental strategic steps that really help, I think you can do that quite efficiently and effectively with less money and less time. And so, and this is the nature of conversations I'm having in other areas too, of how do you describe a scope of work and a project to somebody who wants to give you some money to do something when it's really just the data infrastructure. You're building out like the pipes essentially. And so it's a challenge, but I think there are ways to move forward incrementally, and we're testing that out in California right now with our project.
So I think, Greg, the cost is going to be very different depending on the state and the region. So there's going to be a lot of variability. And it also depends on how the size of the treatment system. Many of the agencies that we work with, they have their own laboratories, and I'm not sure they can actually do the test themselves, or they have the proximity to a laboratory can do the test. So all those things are factors that is going to impact the cost. But I think what is most relevant is really who's paying for it. And in California, we have all wastewater treatment plants are paying for this analysis themselves. So there's not like a direct support right now on this, but it's done on a voluntary effort.
Yeah, and for the 5 and 6 or so treatment plants that we have in California, there's kind of a different data flow in each one of those utility scenarios. And you know, like I know Nathan in Wisconsin, you described the advantages of having, you know, essentially most of the data or at least half the data go through state lab. And so your infrastructure problems and solution sets might look totally different than California's.
Yeah, certainly. Hearing more about how it's operating in different states, it's kind of like, oh, okay, we kind of dodged a bullet there, in that we have had just so few labs and very tight communication between those labs as well. They're always working with each other and talking to try and keep things running smoothly.
Well,
I'll just chime in there on the funding. Just that, okay, my internet might be a little unstable, so just let me know if I freeze. But essentially, so CDC, that since you know we really do view these samples as essentially pooled clinical samples for disease surveillance, differentiating that from sort of environmental monitoring or regulatory monitoring of wastewater or other environmental waters. That essentially makes this participants in this surveillance system eligible for the CDC funding streams. So CDC funds a lot of the surveillance systems around the country using what are called ELC, epidemiology and laboratory capacity building, federal funds. And so news has become part of that sort of annual funding cycle. And so that means that those are monies that go to public health departments. Those are typically at the state level, but there are some sub-state public health departments that qualify for those money as well. So LA County is one of the sub-states. So in California, there's actually at least the state of California as well as LA County both can apply for that money, and that money can be used for epidemiologist staff support for wastewater surveillance. It can be used to support laboratory testing, purchases of laboratory instrumentation, even support for contracting out to commercial testing laboratories. So that is one significant federal funding stream to support this system.
Kyle, I saw you unmute a minute ago. Did you want to chime in on the project cost infrastructure thing?
No, I had some thoughts, but other people have
come. Okay. One of the ideas that I think would be helpful, it seems like, if we could figure out the connection from a LIMS, laboratory information management system, to a ingestion point, whether it's, straight into Decipher's, warehouse or clearinghouse or some other intermediate step, like we're thinking in California, that's that key connection right there. And any kind of extract, transform, load kind of scripts that we could make publicly available. Like if we could sort of, as a minimum, just say, here's an ideal sort of like data format or model. Here's some tools that we would use, knowing that the most common LIMS have these kinds of, data structures already. Those common tools would probably satisfy a lot of the data flow issues. Thoughts?
I mean, to some extent, you're talking about a similar problem that's been hacked away at to death for just environmental water quality monitoring and with the WQX system. And in many ways, the underlying data model of that is pretty much the same thing as what we're talking about. So it might be worth conversing about that at some point later down the line. Understanding, of course, that the WQX system itself doesn't have its detractors, right? But
yeah, I mean, in California, we collect more data that can go into the WQX. So we end up having to sort of create parallel pathways for use of the data in California. But yeah, I mean, that is kind of what I'm thinking, but less rigid about the pipeline. Like, don't enforce that it can only get into the pipeline if it looks like CDC's data. More about sort of like this open innovation kind of approach to allowing different people to sort of like see the code and maybe change it for Nebraska, or did I pick a state that actually isn't doing this? I forget. There was like 2 states that weren't doing this. Yeah. So any other thoughts? Thanks, Kyle.
Yeah, I mean, that's certainly a direction we would like to be moving is, and I think one of the struggles there has just been, that a lot of the labs doing the testing, at least up to this point, are academic labs, or that don't have LIMS systems in place, or historically have never done any kind of formal data reporting before. So I think that as that transition to state health labs happens, or possibly commercial labs with this sort of experience, that will help facilitate that process.
That's great. Thanks, Wiley. Rafa?
So I think it will be perfect if we could actually bring the data directly from the LIMS systems to Decipher. But there's a lot of steps that we need to make sure that there are things that need to happen in that process, in that ETL, which is the data checkers. I mean, the quality control, quality assurance, receiving the data and making sure that it's in the right format with the right values in the right context. And then there has to be some kind of like accreditation, so they can actually, the data is submitted into Decipher with the right user ID and password. I mean, right now is in California, is CDPH who is entering the data or uploading the data into Decipher. But if we come up with a different, more automated process, we're gonna have to, get through all those, privileges and permissions to actually do that. So it's much more complicated than just the ETL. The ETL is probably the easy part.
Yeah. Well, once you know kind of the 2 ends of the equation, right? I'm envisioning like a data exchange, and another metaphor that's been useful in talking in California is this idea of maybe we'll go to like clubs again someday, and there'll be like a waiting area and then a VIP route for anyone who's like going to CDC's Decipher site. Like here's the quick pipeline to go straight into CDC, but there's all this other data that's being captured, at least in California, that probably should come into the club too, and like be at least mixing with some of the other data. So that metaphor is probably about, and I should end that one right here. But what are your thoughts about that, Kyle?
I mean, I think that's a great idea in general, and I'm wondering, this is actually something I want to bring up before, which I'm wondering if in Decipher or in any data system, how much scope there is for a lot of these free text metadata fields for an allowable value to essentially be anything that can be in a URL. The idea being you can link to more data than has maybe been explicitly asked for. And one such link could be to something like what you just described, Greg. Like, here's what we sent to Decipher, but also here's a link to all the other stuff that we collected during the same sampling event or whatever.
Interesting.
So what we may need is like a data warehouse where the data can be put into one place and then from there to be extracted. Some kind of to simplify the process.
Yeah, I mean, that's kind of the California model that's in our head, is to build like a staging warehouse that also would serve as an open data repository for even the poor people that didn't get to go to the CDC party. They'll still be like output, to public data. For the publicly governed data, it'll go to an open data publishing catalog place.
Yeah, we need a place to store the data itself.
Yeah.
And Greg and Rafa, it sounds like you all have worked on these sorts of state level water related data infrastructure projects previously. So what's your experience been like working with other states, or that sort of technology transfer across states, or has it been entirely within California?
Well, my experience has been both, and like one of the better models, in my opinion, even though California's, at least our agency, still isn't participating great, is the WADE 2.0 project for the Western States Water Council. That's Water Data Exchange project. And that data exchange model was actually something that Otto did as his PhD at Utah State. And I got super excited because you essentially could use this web interface, this sort of light look, you're not storing a lot of data there. You're storing a lot of the sort of code that would help people transform their data so they could sort of get informed. They could load their data into this exchange and it would feed back information like, oh, it looks like you have the Ottawa data model. Here's some tools that you could use to transform your data. And you don't even have to route your data through that exchange. It's just sort of like a mirror that feeds back useful information. And I think WADE 2.0 is really moving into that direction, as all the Western states have, as you can imagine, and water rights and water use, we all have different data models and different frequency of collection and all sorts of context issues, right? And so there's a lot of really good stuff happening with that. It's kind of related to this, but and then within other states definitely do this better based on, like Nathan's example of just their data flow and organization structure. It's a lot easier when you have a state lab that wants to work with you and is used to reporting out, and compared to what we have in California, where we have over 2,000 utilities that could be collectors of data, and who knows how many labs and things like that, and academic thoroughfares where the data needs to go first to some other entity before it comes back to the state. So there's all sorts of scenario differences across the country, I think. Nathan, did you want to add anything on that?
No, I was just saying it's been it's been helpful for us to be able to directly integrate with the state labs LIMS system. They can just write things out to us, and then we can sort of curate it, use what we need for our public display, and then send the rest on to CDC. I imagine it would be more difficult to have unless the criteria were very specific, and the labs had the capacity for them to, for every individual lab to just submit automatically to CDC. Because I know even with State Lab, it was we basically took the CDC, the Decipher data dictionary, made a little tweaks for how we needed it and passed it along to the lab saying, hey, here are the data elements we need. Here are the restrictions. And it took them quite a while to actually be able to build that out and to report it. They were just doing CSV extracts to us for quite a while until they were able to build that into their system.
That's really good, useful information. Ashley, do we have any more audience questions?
You do. Okay, so this next one is really about collection protocols. It's a little bit of a change in topic, but is there any guidance available on the actual sample collection protocols? For example, are they point grabs or time series composites? I'm not sure if that's for Wiley mostly or.
Yeah, certainly. There's definitely a kind of general overview of that sample collection process and what those alternatives look like on the new CDC website. So you can certainly, we can share that link afterwards, or I can try to put it in the chat. But so we do have a wastewater surveillance website through CDC that outlines some of those procedures. In terms of some sort of standard collection protocol, that doesn't exist for wastewater surveillance nationally, and CDC doesn't put out standard protocols like an EPA might. But we have been working with APHL, the Association of Public Health Laboratories, to put out some reference sort of protocols. And then I mentioned that CDC, that we're supporting 2 communities of practice. One is for health departments who are implementing this, and the other for laboratories that are working on this sort of testing, public health laboratories specifically, not academic laboratories. The third community of practice that's getting underway now is sponsored and organized by WEF, Water Environment Federation, specifically for utilities, to provide some more of that guidance and support to utilities for sample collection and all of the incredible amount of sort of volunteer work and time that they've been putting into this effort. In general, yes, certainly we want composite samples wherever possible. They are the most representative of what's passing through that sampling point over a given day. Obviously, that's not always possible especially if you go upstream, or we saw these supply chain issues early in the pandemic around auto samplers. But yeah, and where possible, 24-hour composite samples.
And then where needed, metadata description of exactly that so that people can have context, right? Or is that a required field in the actual data elements?
I think it is.
Yeah.
So,
Okay.
so what the key is, like, if it's flow-based or time-based, they have to specify, and what is important is that all the metadata has to be associated with that particular period. So if the composite starts for certain hours, so the PA's conductivity and TSS and the flow reported has to be for that particular period. And I think that's that some systems are struggling with. If they do it the entire day, they say 24 hours for the entire day. So that has to be, all that flow has to be for the day, and everything else has to be, with the same reference.
Right. So it disincentivizes switching back and forth, obviously, because every time you do that, it creates a whole new metadata submission context step, right? Okay, any others?
We have one more quick question. I don't know who this is exactly in best position to answer this question. I'm going to throw it out there for the whole panel, and that is, how does the issue of wastewater volume or dilution relate to the resolution of the analytical methods and the flow proportion reporting be presented?
Anyone can take that if they want. Yeah, I mean, I think the question has to do with like quantifying, and then comparability. So like, obviously dilution is going to vary throughout like time, and it's going to be very specific to that context and time of year because of INI and things like that, right? So there's going to be all sorts of issues that come up, but I think one of the things that I've seen in looking at other dashboards, is like Spain, for example. Madrid has a really good dashboard showing just colors of sewer sheds based on up or down or staying the same, I believe, and then shades of color depending on how fast it's changing in a direction. So you could, potentially still use data that has a whole different kind of dilution context, even though it's collected differently from the other data sets. So.
We have been exploring some different methods of data normalization, like Pepper mild model virus, some of those different things that should, in theory, fluctuate with dilution as SARS-CoV-2 does, but we haven't, those have just been for internal analyses. We haven't done anything that is public facing just because lack of confidence in it at this point.
I recall Utah was doing it too.
I think at the end of the day, sort of the value that you're interested in is being able to count what's the total number of these SARS-CoV-2 RNA particles that are passing through my sampling point at any given point in time. And so you need, and then sort of relating that to what the population is that's contributing wastewater to that point. And the idea is hopefully that gives you some sort of representation of the disease burden or the infection burden in the community. And so you need some way to get from the concentrations that you're measuring to that count. And, I think using some sort of endogenous control like a Pepper mild model virus is one way, using flow is another way. In some situations, it seems it's very important when those variables vary quite a bit. In other situations, it seems perhaps like it's a little bit less variable, and the wastewater content is quite stable over time. But that's going to vary from the fraction, whether you're having INI, whether you have large industrial or commercial inputs that vary on a weekly or sub-weekly time scale. So certainly some sort of evaluation certainly needs to be done, and then that those other metadata around population and around either a flow or some sort of endogenous control are really important.
And I think that's all, Greg. We have a few things to wrap up for the peer-to-peer network. Did you have any final comments or questions?
I want to thank you, the Internet of Water, and the panelists. I thought it was actually a really interesting conversation. I hope other people found it interesting too, but I'm glad it's recorded and I might refer back to this. But thank you, panelists, every one of you.
Thank you.
Thank you.
Yeah, thank you.
Thank all of you as well. And I invite you to continue the conversation or submit additional questions to our panelists at the peer-to-peer forum, which you can locate at the link here on the slide. We also have a quick a poll that we'd like to invite you to complete. If you please take a moment or two and complete this poll, that would be fantastic. Otherwise, again, we would like to invite you to join our peer-to-peer network. You can go to our webpage and internetofwater.org, and you'll see at the top of the webpage a menu item for the peer-to-peer network. I hope you'll consider visiting that site and registering for our network. And we thank you all for your participation. Again, the video to this webinar will be available on our website and will be emailed to all of you who registered. Thank you so much, and we hope you have a great rest of the week.